Hi everybody. In this video lesson, we are going to finish up our unit on biotechnology and genetic engineering by looking at the Human Genome Project and what's often called DNA fingerprinting, something you might have heard of. The Human Genome Project is just what the name says. A genome is like a library. So just like you could go to Barnes & Noble's and there's a whole section just on cookbooks, and they're usually in alphabetical order by, either the name of the cookbook or the name of the author, just like in the library. Well, the Human Genome Project, we actually have a library of genes, and that's what we call the genome. And this project is an international effort to completely map and sequence the human genome. Basically, it'd be like taking a piece of human DNA and opening it up page by page and copying every single recipe down in order to try and figure out what does that gene makes. Now, our human genome consists of about 35,000 to 40,000 genes. Now, these are what we call expressed genes. These are genes that actually get turned on and get used. But some of our DNA actually can have up to 80,000 genes in it. But most of them might be vestigial, genes that we don't use anymore, but nature hasn't gotten rid of just in case we need it again, or maybe like some junk genes. Once we figure out the Human Genome Project and we figured out what different genes are, we can do what's called a linkage map. So just like you look at a map to try and figure out where a street is or where a road is or where a building is, we can use linkage maps to show where a certain gene is. So linkage maps shows the locations of genes on chromosomes. If you take AP biology, you're going to go into the human genome and linkage maps in a lot more detail. You'll learn what the white regions mean, what the black regions mean, and what we mean by linkage. But for basic high school biology, just know that linkage maps, just like a map shows you where things are found, linkage map shows us locations of genes on chromosomes. So for example, we know exactly what the insulin gene is, so we can look on the map and we can see, okay, this is where the insulin gene is found on human DNA. Why do we want to do this? Well, because this will help us diagnose certain genetic disorders. Remember, when we look at mutations, you have two types of mutations. You have a gene mutation, something like diabetes, where just one recipe gets messed up. Or you can have a chromosomal mutation, something like Down syndrome or Turner syndrome, where an entire chromosome is either repeated or deleted. So by knowing where certain genes are on each chromosome, that will help us diagnose whether or not somebody has a genetic disorder. It will also allow us to do something called gene therapy. Gene therapy is where we insert a normal gene in order to correct a disorder. Now, this is not a cure, it's a therapy. So for example, here, one of the most famous types of gene therapy deals with cystic fibrosis. Cystic fibrosis is a genetic disorder which causes a large amount of mucus to build up inside of the lungs and air passageways of people affected by this disorder. We know what gene causes cystic fibrosis. So what if we could take a virus like the flu? The flu virus infects your lung tissues. And what if I could genetically modify that virus? Basically what that means, when a virus infects you, it takes over your cells and it causes your cells to make baby viruses. And then your cells explode open and all these baby viruses come flying out. Well, what if I could take the flu virus and genetically modify it so that it will still infect your lung tissue, but it won't do anything bad to your lung tissue? And at the same time, what if I could take the human gene for not having cystic fibrosis and insert it into this person's, into the virus? So now when the virus infects the person with cystic fibrosis, it's not going to make them sick, but it will temporarily help them. It will temporarily stop them from creating all this extra mucus. Something else I can do now that we have an idea of how DNA works is we can do what's called DNA fingerprinting. You've probably heard about this on like crime shows and different movies. Well, just like our fingerprints are different, everybody's DNA is a little bit different. And so we can actually come up with what we call DNA fingerprints. And it's basically segments of DNA inside of a gel. But I'm going to show you guys how to read it. DNA fingerprinting uses something called gel electrophoresis. And Pore means openings, and electro is dealing to electricity. And so this concept of gel electrophoresis is used to create a DNA fingerprint. And since all of our DNA is different, everybody has a unique pattern of DNA fragments for each person. Basically what I would do is I would take this piece of DNA and I would cut it into small pieces using restriction enzymes, our little scissors. And since my DNA has a little bit of a different pattern than your DNA, the restriction enzymes are going to cut the pieces a little bit differently. It would be like me having a bunch of string, and I cut one piece of string maybe every 5 inches, and I cut another piece of string maybe every 4 inches. So even though it's the same kind of string, just based on how it's cut, I'm going to get different size lengths of string. This is what happens with DNA fingerprinting. So DNA samples can be taken from any cell that contains DNA. It's most commonly taken from blood samples, hair, semen, spit, or even skin tissue cells. I can then isolate that DNA and use restriction enzymes to cut it. And again, because everyone's DNA is different, one restriction enzyme will cut different people's DNA in different places. So like I said, it would be like me using different lengths of, or using a length of string, and I cut one piece of string every 5 inches, and I cut another piece of string every 4 inches. That's going to give me different lengths pieces of string. Well, this is what happens with our DNA. Restriction enzymes are going to cut our DNA into little different pieces. Well, every piece of DNA is going to get cut a little bit differently. So my pieces of DNA will be different lengths than your pieces of DNA. We then insert it into what's called a gel. This is why it's called gel electrophoresis. And so that's what you're seeing here in the screen. It's very much like Jell-O, and it has what we call wells on it. Okay? These little things right here are called wells. And that's where I will insert that person's DNA. So basically, I put your DNA into a test tube, add a little bit of liquid to it, put those restriction enzymes in there, let the restriction enzymes cut the DNA up. Then I take a pipette and I insert it into this little well. Well, DNA has a charge to it. DNA actually has a negative charge, and you know that like charges repel and unlike charges attract each other. So what we'll do is we basically hook this up to a battery or hook this up to an electrical charge of some type, and I let electricity run through the gel. And as electricity runs through the gel, DNA, as you can see here, gets pushed down through the gel. And the longer the piece of DNA, the harder it is to get pushed through. The smaller the piece of DNA, the easier it is to get passed through. So think of it this way, you have to push rocks around. It's really easy for you to push a pebble, but it's really hard for you to push a boulder. Well, we use electricity to kind of push DNA through this thick gel. The longer pieces of DNA will stay at the top, and the shorter pieces of DNA will move down towards the bottom, which allows us to have a DNA fingerprint. Since nobody's DNA is cut the exact same way, nobody's DNA is going to end up in about the same pattern. So gel electrophoresis is going to separate these segments by size. Now remember, these segments don't tell me what the gene is. Like I can't look at this and tell, is this AAA, GGG, you know. So what I mean by that is I can't look at this piece here and tell that it's AUG, or excuse me, ATGCCA. I just know it's so long. So we can use this, for example, crime scenes. So if you take a look at this gel electrophoresis, here's my victim, and you can see that my victim's DNA matches blood that was found at crime scene number 2. So maybe there was a little bit of blood splatter, and a person came to the hospital and they just weren't sure what crime scene it was. Then I have different suspects. Well, notice here, suspect 2 had nothing to do with the victim, but suspect 2 was at crime scene number 2. And then suspect number 1, if you'll notice, doesn't match up with anything. So suspect number 1 had nothing to do with either of these crime scenes. So just a shorter version of how DNA fingerprints are made. Keep in mind that DNA has a negative charge. You put the DNA in the gel, and when you turn on the gel, electricity runs through it. So Unlike charges attract, so the negatively charged DNA is going to be attracted to the positively charged side. So that's going to basically push the DNA samples through the gel. So because the gel is porous, large fragments get stuck and don't move as far, while the smaller fragments, like pushing up pebbles, can go further. So when you interpret a gel, keep in mind, most of the time, ignore the top row, because those are those wells. That's where I put the DNA to start with. And then I hook it up to a battery. So I have, just like a battery has a negative side and a positive side, and I let electricity run through, and the DNA is going to move down. So the DNA at the top is going to be longer than the DNA down at the bottom. So let's say that, again, we have this blood stain, and we need to figure out who this blood belongs to. So notice with your first one here, okay, it matches up with Bob, John, and Lisa. Lisa's a little bit down below, but it's still a match. So we have to move on. But if you'll notice here with my blood sample, Bob is now out of the running. However, it says that it matches up with Sue, but it doesn't match up with Sue right here. And if you continue your way down, you can see how this blood sample matches up with John. I'm just going to stop circling. John all the way down. Here's another example of a gel that was used in forensic science. There was a semen sample, and there were 2 victims. And you can see here how this sample came from this suspect because it matches up. It can also be used in paternity testing. Now, when you do paternity testing, you have to have a sample of the mother and a sample from the father. Because remember, as a child, you get half the information from your dad, you get half the information from your mom. So let's take a look at this one here. We have a mom and we have a dad. Notice daughter number 1. She has some genes that match her mom, and she has some DNA that matches her dad. So daughter number 1 is a biological child. Notice daughter number 2. Daughter number 2 has DNA that matches her mom. But notice she also has DNA from a different source. Okay? Let's look at what's in red. So it kind of looks like it matches here and here. But notice this one does not line up like it's supposed to. So maybe this is a daughter from a former marriage. Okay? The daughter does belong to the mom, but it does not belong to this father. If you look at son number 1, he has DNA from his mom, he has DNA from his dad. And if you look at son number 2, he does not have DNA from either parent. Nothing matches up with the mom, nothing matches up with the dad. So maybe this is a child that was adopted, or maybe is just living in that home. So when you're interpreting a gel, when we look at every band in paternity testing, in the child, either has to show up in the mom or it has to show up in the dad. So if you'll take a look at this one right here, okay, here's the child. And notice this piece of DNA matches the dad. This piece of DNA matches the mom. So when we do paternity testing, it has to line up. Half the DNA from the dad, or when you look at the kid's DNA, some of it has to line up with the mom, some of it's going to line up with the dad. So for example here, we have Mary, Bob, and Larry, and the child. You can see how the child got this part of its DNA from the mom. However, this part of the DNA came from the dad. So Larry is the father of that child. So we can use DNA fingerprinting in paternity testing. We can use it in criminal investigations, like I showed you at crime scenes. We can also use it to help identify fossilized remains. So if a body is found, we can take, if there's DNA available, we can take DNA and compare it to different family members and see if there is a match in order to help identify victims. Sometimes, however, there's not enough DNA to do these tests. So like we might find a fossil and we can just get the barest amount of DNA. Or we might find a piece of bone, and we're trying to identify if this is a missing person. So there's not a lot of DNA available. Well, we can do what's called polymerase chain reaction. Basically, let's go back to making a transgenic organism. What if I take that small piece of human DNA and put it into a bacteria, and I just let that bacteria divide? Over and over and over and over again, that bacteria divides. So that bacteria is replicating this human or whatever DNA that I want to test. And then when I have enough of it, I can go in, I can cut that DNA out of the bacteria, and then I can do whatever test that I need to run on the DNA. And if you ever get a chance, you can Google A Criminal in the Family, a CBS News. It's from 60 Minutes. It's a really interesting way, shows you really interesting ways that we're using DNA in order to solve crimes.